Hello everyone, this is Amy. In this video, we'll briefly talk about gene expression and protein synthesis. Synthesis of proteins is an important function that the cells perform in our body. We've discussed earlier that a segment of a chromosome is a gene that carries instructions to code for a protein which our body needs. Gene expression is simply the process that converts instructions from a gene into a protein. Let's just think for a moment. DNA stays in a cell's nucleus, but protein synthesis occurs in the cytoplasm. How does that happen? How do the instructions get transferred outside of the nucleus? Well, protein synthesis occurs in two steps: transcription and translation. In transcription, the instructions get copied from DNA and are coded into RNA. RNA is another nucleic acid found in our body. RNA stands for ribonucleic acid, and it's single-stranded, unlike DNA, which is double-stranded. RNA is much shorter than DNA, and hence it can exit the nucleus with the instructions. A special type of RNA, called messenger RNA, or mRNA, helps in the process of transcription. Once mRNA reaches the cytoplasm, the instructions get translated into a protein. We'll talk about both in detail in a moment. Let's first spend a few moments discussing both nucleic acids, DNA and RNA. DNA and RNA differ from each other as shown here. We've already said that DNA is part of chromosomes and is in the nucleus, whereas RNA is found in the cytoplasm. DNA contains deoxyribose, while RNA has ribose. Notice their structures. Everything else is the same, other than this H for DNA versus this hydroxyl or OH group for RNA. We've also said that DNA is double-stranded, whereas RNA is single-stranded. Function-wise, DNA carries genetic information or instructions for protein synthesis. DNA also replicates prior to cell division. In contrast, the main function of RNA is to help with protein synthesis. There are various types of RNA as shown here. They are messenger RNA, transfer RNA, and ribosomal RNA. Finally, both DNA and RNA have 4 nitrogenous bases, and 3 are the same for both. Those 3 are adenine, cytosine, and guanine. While DNA has thymine, however, RNA has uracil. The complementary base pairs of DNA are shown here. A recap is that adenine always bonds with thymine and vice versa, whereas cytosine always bonds with guanine and vice versa in DNA. However, there is no thymine in RNA, so anywhere thymine is needed, it will be replaced with uracil in mRNA during transcription, as shown here. That's how the instructions from DNA get coded or transcribed into mRNA. Let's now look at the transcription process in a bit more detail. The enzyme RNA polymerase synthesizes mRNA following complementary base pairing, as we've just discussed. RNA polymerase also knows where the gene in the chromosome begins. This region is called the promoter. RNA polymerase also knows where the gene ends. This region is called the terminator. RNA polymerase also knows the right direction to read the DNA. First, RNA polymerase pulls apart the DNA strands. Only one of the 2 strands is transcribed, and that strand is called the template strand. The other strand is called the coding strand. There are 3 phases of transcription: initiation, elongation, and termination. During the first phase, initiation, RNA polymerase latches onto the template strand at the promoter, or the starting point, and starts transcribing, or coding, the complementary base pairs. Let's look at another image to understand this better. This is the template strand. This is the coding strand. And this is the direction in which coding is being performed. Notice that the first base here on the template strand is thymine. Its complementary base is adenine, as it can be seen on the coding strand, so the mRNA also codes adenine. The next one is cytosine. Its complementary base is guanine, so the mRNA codes guanine. The next two will be coded in a similar manner. Let's look at this fifth one. This is adenine on the template strand, and its complementary base is thymine. However, RNA does not have thymine, so it will code uracil there for adenine. The rest of the bases will be coded accordingly, as we've discussed, until RNA hits the terminator. This is how the transcription unit elongates, and this stage is called the elongation stage. The final stage of the transcription process is the termination stage. Once RNA polymerase hits the end, it stops coding and releases the transcribed mRNA or the transcription unit to the cytoplasm. This marks the end of the transcription process. Let's look at an example of transcription. Let's say we're looking at a DNA template strand that has this sequence. The DNA complementary base pairs for this sequence on the coding strand would be thymine and adenine, adenine and thymine, cytosine and guanine, adenine and thymine, guanine and cytosine, again guanine and cytosine, another guanine and cytosine, adenine to thymine, cytosine to guanine, adenine to thymine, and then thymine to adenine, and another thymine and adenine. The corresponding mRNA strand would be adenine, stays like that, thymine becomes uracil. Remember, there's no thymine in RNA. Guanine stays as guanine, thymine again becomes uracil. These two cytosines stay the same. The cytosine stays as a cytosine. The thymine becomes uracil. Guanine stays itself. Thymine becomes uracil, and then the two adenines stay as themselves. Next, translation. So far, we've said that translation occurs in the cytoplasm, specifically in the ribosome. Once the DNA instructions get coded or transcribed in the nucleus, it is then decoded or translated in the cytoplasm, and then the protein is formed. We know that the building blocks of a protein are amino acids. Translation can be viewed as a translation from nucleic acids to amino acids. Each amino acid can be seen as a sequence of 3 contiguous bases in the DNA sequence. The 3 bases are called codons in mRNA. Going back to our example, this is a codon, this is another, and so on. Codons denote specific amino acids used to make proteins. Let's look at the mRNA codon chart that helps with translation. Notice that AUG, also called methionine, denotes the start of an amino acid or gene. There's just one start with 3 stops to indicate the end of a sequence or a gene. The mRNA, after leaving the nucleus, attaches to a ribosome in the cytoplasm. There, another type of RNA, called transfer RNA, or tRNA, plays an important role. Let's spend a few moments on tRNA. Just like mRNA, tRNA is also synthesized in the nucleus and is sent to the cytoplasm to help with the synthesis of the protein. tRNA, or transfer RNA, is the translator. The 3 nucleotides on the tRNA are called an anticodon. tRNA brings the correct amino acid from the pool of amino acids in the cytoplasm and adds it to the growing protein or polypeptide chain on the ribosome. After a chemical bond holds 2 adjacent amino acids together, tRNA is released from its amino acid and is recycled. As the protein molecule forms, it folds into its unique conformation and is released to be used by the body. Correct folding of proteins is essential to our health. Misfolded proteins are either refolded into their correct conformation or are destroyed if they're not correctable or too abnormal. Let's now revisit our earlier example. So our mRNA strand has this sequence. Following the codon chart, we can see that AUG over here is methionine, UCC is serine, CUG is over here, leucine, and UAA is the stop codon. Let's recap the process. This is the template strand in DNA, the strand to be transcribed. This is the coding strand in DNA. This is the mRNA strand that is transcribed. And this is the polypeptide chain, or the protein, that is formed after translation is complete. Finally, in a nutshell, protein synthesis can be viewed as this. DNA's instructions get transcribed into RNA, which then gets translated into the needed protein. When a gene is transcribed and translated into a protein, it is said to be expressed. That was a brief overview of protein synthesis. Thank you for watching.